My name is Parker Bogus. I'm an undergraduate researcher in the Tissue Engineering and Regenerative Medicine Laboratory at Northern Arizona University in Flagstaff, Arizona. That's in the United States. My undergraduate research, another project I did was an in vitro glioblastoma assay to demonstrate the effect of 2-deoxy-D-glucose and metformin as a synergistic treatment option. I did this with my research partner, Bryce Jewett, under Dr. Robert S. Keller. So the societal relevance of glioblastoma multiforme, also known as GBM, it is an aggressive and invasive grade 4 glioma. It's the most common type of brain cancer found in adults. It's often located in the frontal and temporal lobes of the brain. It unfortunately affects 3 individuals per every 100,000 annually in the United States, with incidence rates only continuing to rise. The standard treatment protocol for glioblastoma multiforme is called the Stoop protocol, which is the surgical resection of the tumor followed by subsequent bouts of radiation and chemotherapy. It's important to note it's quite difficult to completely remove the tumor via surgery. Interestingly, the protocol has actually remained unchanged since 2005, and due to this, the GBM has an extremely high recurrence rate and a median survival time of only approximately 6 to 12 months, which prompts new therapeutic methods to be developed. So it's important to understand metabolism and also cancer cell metabolism in order to understand what we are trying to accomplish with our project. So in a healthy cell, when there's oxygen abundant, glucose will come into the cell, undergo a series of 10 steps known as glycolysis, which forms a product known as pyruvate. This pyruvate is then bridged into another product called acetyl-CoA, and acetyl-CoA will undergo what's called the Krebs cycle, and the Krebs cycle harvest electrons, and these electrons then go through a series of steps in the electron transport chain, which yields water, but more importantly, it yields a lot of ATP, which is the cell's energy currency. And it's so that's a very efficient process. So this is a healthy cell when oxygen is abundant. So still in a healthy cell when there is not an abundance of oxygen. So if you can think of a situation where there's not an abundance of oxygen, that would be like if you're doing a full out sprint or something of the sort. Glucose still comes into the cell, undergoes the series of steps in glycolysis, which forms pyruvate. But instead of pyruvate being turned into acetyl-CoA and subsequently undergoing Krebs and electron transport chain, pyruvate is then converted into lactate. Now, contrary to popular belief, in most scenarios, lactate is actually extremely beneficial for a long period of time. There is a common notion that was incorrect that lactate and lactic acid specifically was bad for you, but it couldn't actually be farther from the truth in healthy cells. Now, in cancer cells, when there's oxygen abundant, glucose enters the cell, of course, undergoes glycolysis, which turns into pyruvate. So what do you think happens next? Do you think pyruvate is turned into lactate, or do you think pyruvate is turned into acetyl-CoA, subsequently undergoing Krebs and electron transport chain? Well, logical thinking would actually lead you to believe that the latter would happen and that it would go through Krebs and electron transport chain. However, there is a phenomenon called the Warburg effect in cancer cells, in which even in the presence of ample oxygen, pyruvate is converted into lactate. And in this scenario, lactate is extremely harmful. So an increase in lactate it being produced by the cells is actually transported out into the tumor microenvironment, which can contribute to a variety of the hallmarks of cancer. And the hallmarks of cancer are series of characteristics that every single cancer cell possesses, including glioblastoma cells in the brain. Some examples of the hallmarks of cancer induced by lactate or hypothesized to be induced by lactate include the evasion of our immune system. So immune system cells not being able to kill these cancer cells. Angiogenesis, which is the formation of new blood vessels. So this lactate is causing angiogenesis. So new blood vessels means that the oxygen and nutrients are getting to these cells, which is not good because they can therefore proliferate even more than they already are. And then most deadly is metastasis, where a certain type of cancer in one part of the body is able to migrate into the bloodstream and go and travel to other parts of the body. So 2-deoxy-D-glucose, 2DG for short, it's very important to understand its mechanism of action. So here you can see, similar to what we just described previously, glucose comes into the cell via a receptor. This receptor is specific to glioblastoma cells in the brain. So it comes into this GLUT3 receptor, and glucose undergoes glycolysis. So glucose is converted into glucose 6 phosphate by an enzyme known as hexokinase. However, 2-DG actually inhibits this step because 2-DG is a glucose analog. So it inhibits the step, which the downstream effects means that there isn't a lot of lactate being produced. Therefore, the lactate can't be transported into the tumor microenvironment. And these hallmarks of cancer can't take place to the extent that they could if this step was occurring and there wasn't an inhibition via 2-DG. So it's important to note that a majority of these cancer cells, they're cancer cells in the tumor core, they're hypoxic. Hypoxic meaning that there's not an abundance of oxygen. That's why they're preferring to undergo glycolysis. And that is the Warburg effect in essence. However, not all cells are undergoing the Warburg effect. Some of these cancer cells are still undergoing the full Krebs cycle and electron transport chain. To inhibit these cells, metformin inhibits an important part of the electron transport chain known as the mitochondrial complex 1. And what this does is it increases the ratio of adenosine monophosphate, AMP, to adenosine triphosphate, ATP. And in essence, what this is telling the cell is that, oh my goodness, there's a low energy environment, so we need to conserve energy. So we don't have time to proliferate and go and divide right now. We need to save energy so we ourselves don't apoptose and don't die. So this increase in AMP to ATP ratio triggers what's called as the AMPK pathway, and the AMPK pathway is notorious for inhibiting the mTOR complex 1. The mTOR complex 1 is extremely important for not only protein synthesis, but then subsequently cell growth and cell proliferation. So you've got metformin, which causes downstream effects that tells the cell, oh, we can't divide and grow right now. So combining 2-DG, which is a glycolysis inhibitor, so most of these cells are undergoing glycolysis in the Warburg effect. So 2-DG is inhibiting most of these cells from proliferating, but then the cells that are still undergoing the Krebs and subsequently electron transport chain, metformin is inhibiting them through AMPK signaling and through the inhibition of mTOR complex 1. So how we conducted this experiment, we seeded these cells into a 96-well plate. After we grew the cells to confluence, we subsequently added the respective treatments. So we did 2-DG as a monotherapy, metformin as a monotherapy, and then lastly 2-DG and metformin as a synergistic therapy. So we did 3 different trials of this. So following the addition of the respective treatment, we added the addition of the Presta Blue reagent. We followed an incubation step, which caused a colorimetric reaction to assess metabolic activity. And we measured this via an absorbance reading through fluorescence. After this reading, we added an additional reagent called Cyquant and followed an additional incubation period, which again caused a colorimetric reaction. So we were able to measure the absorbance to assess the DNA content of these cells. So not only did we assess metabolic activity, but we also assessed DNA content. So these first few images are 2-DG as a monotherapy. So it was 2-DG by itself. And the figure on the left is the metabolic activity of 2-DG. And as you can see, with increasing concentration, the metabolic activity follows in suit and decreases. This can be seen also in the DNA content. So increasing 2-DG concentration, decreased metabolic activity. And so we did a metabolic activity assay via Presta Blue and DNA content via a Cyquant assay, but to find metabolic activity, we took the fluorescence that we got from the metabolic activity and actually divided it by the DNA content, yielding a more accurate depiction of what's actually occurring inside the cell, so giving us this metabolic index. So you could see that, again, with increasing concentration, the metabolic index also decreases. Moving on to metformin as a monotherapy, again, figure on the left is showing the metabolic activity, increasing concentrations of metformin, decreased metabolic activity. Same with DNA content, increasing concentration, decreased DNA content. However, interestingly, when we did the metabolic index, which again is the metabolic activity, the fluorescence obtained through the metabolic activity divided by the fluorescence obtained from the DNA content, we actually found that the 2.5 millimolar of metformin treatment was the most effective at inhibiting metabolic index. Due to this finding, we decided to keep the 2.5 metformin treatment consistent throughout, and we only changed the concentration of 2DG. So these three figures are showing 2DG and metformin as a synergistic therapy. So we're using these treatments together. And so we did varying concentrations of 2DG, so 4, 6, 8, 10, but we remained constant with that metformin from the previous finding of 2.5 millimolar. So again, metabolic activity, it decreased with increasing concentration of 2DG. However, the 4 millimolar was not necessarily different from that of, although it was statistically significant to that of the control, it was not statistically significant to that of higher concentrations. So although visually you can see that the 10 millimolar 2DG, 2.5 millimolar metformin had the lowest metabolic activity out of the treatment groups, it was not significantly different from that of the lower dosage of 4 millimolar 2DG, 2.5 millimolar metformin. Similar trends can be seen in the DNA contents of the 2DG and metformin as a synergistic treatment. So again, starting at the 4 millimolar 2DG, 2.5 millimolar metformin, you could see statistical difference from that of the control, but not necessarily from that of the highest concentration, that being 2 millimolar 2DG, 2.5 millimolar metformin. The metabolic index also suggests this. So the 4 millimolar 4 millimolar of 2DG, 2.5 millimolar of metformin was statistically significant to that of the control. But it was only statistically significant to that of the 10 millimolar 2DG, 2.5 millimolar metformin. So what we concluded was that combining the 4 millimolar 2DG with the 2.5 millimolar metformin significantly inhibited glioblastoma cell growth via inhibiting metabolic activity and DNA content while maintaining a favorable metabolic index, indicating effective tumor suppression with minimal cytotoxicity to healthy cells. So if we go back here, if you were to use this treatment, even though it had the lowest metabolic activity out of any of the tests that we ran, this 10 millimolar 2DG and 2.5 millimolar metformin, if used in the actual patient, it would likely be extremely cytotoxic, not only to these glioblastoma cells, but also to healthy cells. And that's why we believe that the optimal concentration is the low dosage of 4 millimolar 2DG with the 2.5 millimolar metformin. So to confirm the underlying proposed mechanisms that I discussed earlier, future in vitro studies should include Western blots looking at the AMPK protein and the mTOR protein, and seahorse assays to determine the ATP or energy production of these cells to further evaluate this optimized low dosage combination. After comprehensive in vitro validation following these two assays, this synergistic treatment should be translated to in vivo animal models using an impregnated scaffold for localized delivery. I'd like to acknowledge my research partner, Bryce Jewett, our research mentor, Dr. Robert S. Keller, and the other members of the NAU Term Lab for their help on this project. I'd like to acknowledge the NIH grant, which Dr. Robert S. Keller received in order to help fund this project, Development Engineering Sciences for providing additional funding for this project, and Barrow Neurological Institute for generously providing the U118MG glioblastoma cells that we used for these assays. Here are the references that we use for our project. Thank you for listening and have a great day.